In this section of the video, I'm going to show you how to use the blood typing kit. So we're going to be using synthetic blood, not real blood, so there's no risk of contamination or anything. There's no special cleanup procedures, but they come in kits like this. And inside, it's got some stuff. We have, as you see, we've got some wet erase markers. You have what we call the blood typing slides that have 3 wells in them. They're like dishes, and there's 3 different colors of stirring sticks, and you'll also find the bloods, or the synthetic bloods, and the anti-sera that cause them to coagulate. So we have 4 different types of blood, and they are labeled with names, and what you're going to be doing is testing some paternity of a child. So you have Andrew, Jack, Daniel, and Megan. So I'm going to pick a random blood type to do. I don't know which one it's going to be. I don't know what it's going to look like, but I'll show you the procedure. So what you want to do is gather your blood typing slide. And now I know it's a little bit hard to see, but if you look carefully, you can kind of see through here and see that it says RH really faintly. See if I have something dark to put it up against. Knock everything off. Let's see if this helps. Yeah, there we go. So you see how it says really faintly right there, RH? We have A and then B down here at the bottom. So what I'd like you to do with your marker is first write over those and write exactly what they say. So I'm upside down, so I'm going to do it this way. RH, A, and B. This is just going to prevent any confusion when you're working on your actual blood typing. So then what I want you to do is down at this corner, where you don't have anything written, to write the initial of whoever you're testing. So I'm going to do X because I don't know. We're just going to do that. So if you're doing Megan, do M. If you're doing Daniel, do D, etc. So the first thing you want to do is pick your blood sample. So I'm going to pick one of these randomly, so you won't know what it is. I'm going to shake it up. Shake it really well. And then I'm going to put 1 drop in each of these wells. One drop of blood, fake blood. Okay. Our next step is to add our anti-sera. So these cause them to coagulate when they interact with whatever is in here. So if I'm going to add the anti-A serum, there it is, anti-A. So I'm going to add 1 drop in the well that says A. Now make sure not to touch the surface here. You can hold it up above. It's fine. So add 1 drop, we're in A. We're going to add 1 drop of anti-B, which is yellow, where it says B. And then 1 drop of anti-D, where it says RH. I know it's confusing, but same thing. All right, there we go. So what I'm going to do here, I'm going to take my color-coordinated stirring sticks, blue for A, yellow for B, and white for anti-D. Oh no, there's a cat. She wants to come onto the desk. Not a good time. So I'm going to stir up A. And I'm going to stir for about 15 to 20 seconds, and look at what's happening here. Oh, I'm splattering because I'm not watching. So please don't be me. Don't do that. That's why one of the supplies is a paper towel. So notice here that you see a change. You see it clumping together. This is called agglutination, and this means that the anti-A serum is interacting with A antigens on the surface of these fake blood cells. So I hope you can see that okay. So you might have to tilt it a little bit to see. But there it is. The focus is really not wanting to work. You gonna work? A little bit, sort of. There we go. The lighting is also not great in here. So you're going to continue to do the same. Discard your sticks. Do not reuse them. There's a trash can there. And now I'm going to stir B. So stir, 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 stir. about 20 seconds. Now some of these are more obvious than others, so you might have to do some looking closely. What I'm seeing here is that there is actually some agglutination. Let's see if it'll focus. Come on, I know you will. I know you're capable. There we go. So we actually see here, little platform. We actually see some little bitty clumps here, but it's definitely clumping. See them there? All right, so I'm going to stir up here. Now let's stir my RH factor here. Stir, 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 stir. Now I am not seeing anything happening here. It just looks like one solid color with no clumping or anything. Not seeing anything. You see how different they are? How you've got pretty clear clumps, but also really no clumps in the RH factor. So this individual's results, they have A antigens, B antigens, but no RH antigens, which means this individual is AB negative. So if it does interact, then they do have that antigen. I know it's a little bit confusing, but that's how it works. All right, so here's how to actually do this. And the next and final part of the video, or final video, whichever I decide to do, separate or together, will be explaining how to figure out the genotype based on this. Thanks for watching.